In your experience, how much would you say that telomere length, so the telomeres get shorter with time, and shorter telomeres are supposed to correspond to aging. How much would you say that telomere length regulates the aging process, like actually plays an active role, versus just is a biomarker, something that's just biomarking the aging process?
Yeah, that's a good question. So telomeres are one specific pathway of how a cell ages and how our tissue ages. And the pathway is this. It's called replicative senescence, and it's basically how long can that cell continue to divide and replenish into new, fresh, young cells. So the telomeres, when they get too short, prevent that particular cell, whether it's an immune cell or a neuron in our hippocampus or the lining of our cardiovascular system. We need those cells to replenish throughout the decades. When the telomere gets too short, that cell stops dividing. And so it's basically a little window into how long can these cells continue dividing. If the telomeres are long, they have a long potential for replenishing tissue. One last point about genetics. So you earlier asked, is this a marker of aging or is it a mechanism?
Right.
So it is probably both. And the way we know that it's the mechanism as well is, partly the example of some of these genetic disorders, but even more so, now we know that if you have a genetic propensity for long telomeres, it directly predicts less heart disease and dementia. So that is, those kind of Mendelian randomization studies are one of the best ways that we can say there's a direct physiological connection here.
Right, yeah. I didn't know there was any Mendelian randomization studies on that. That's very interesting. So cardiovascular disease and dementia are two
Those are
health outcomes that seem to be affected.
Right, and then as I said, the higher cancer risk for some of these.